Hello, my name is Zachary Abdullahi, and I am in Bio 402. And today, for this lab, I'm going to be making apple cider vinegar. Cells under anaerobic conditions need to utilize fermentation in order to recycle the excess NADH produced by glycolysis. To do so, they will convert NADH along with pyruvate into other products and also creating intermediates such as CO2, alcohol, and other chemicals. In this lab, yeast and bacteria will be used to ferment apples and sugar. Yeast will ferment the sugar into alcohol. That alcohol will be later fermented by the bacteria into acetic acid, which will produce the vinegar. The ingredients for this lab include apples, some water, 2 glass jars, coffee filters, sugar, apple cider vinegar containing a mother, apple cider vinegar to serve as a positive control, and 3 pH strips. First, we'll begin with the apples. Separate the apple core from the rest of the apple. The apple cores contain seeds which have inorganic arsenic and cyanide, which we don't want to include in the apple cider vinegar. Once the apples are cut, you want to start placing them into each jar. For this experiment, I used a total of 3 apples. Keep filling the jars until each jar is filled up to 3/4 of the way with apples. Next, the starter is added. The mother, which is in the apple cider vinegar, contains yeast, bacteria, and proteins. These will serve as a starter which will kickstart the fermentation process. Half a cup is added to the sample only and not the negative control. The sugar serves as food for yeast, which we turn into alcohol. Half a cup is added to both the sample and negative control. Next, warm water is going to be added to both jars to fill up to the very top and to make sure that the apples are completely submerged. Then we're going to stir it up to make sure that the sugar is completely dissolved within the water. Next, both the sample and negative control are going to be covered with a coffee filter with a label to denote which is which. They're both going to be placed into a dark, warm area for 6 days. 6 days are now up, and it's now time to look at our apple cider vinegar. By looking at it, it's hard to tell if there's any difference between the bubble formation between our sample and the negative control. I think this is because the jars were not fermented long enough to have a significant difference. When looking at the apples from the negative control and the sample has a darker tone compared to the negative control. This is evidence that there must be more fermentation going on in the sample compared to the negative control. Also, looking at the pH strips, the sample has a lower pH compared to the negative control, meaning that there is more fermentation going on, producing higher amounts of acetic acid in the sample compared to the negative control. Lastly, I'd like to thank apple farmers all across the world for producing quality apples like the ones used in this experiment. Thank you for watching, and I hope you enjoyed.